Hey, Reef Builders, welcome back to another session of Reef Therapy. My name is Remy. I'm joined by Salem Clemens, Dr. Eli Meyer, owner of Aquabiomics, and also Dr. Andrew Bouwma, both of you guys are in Oregon, right? Dr. Andrew Bouwma, you're working for Oregon State, but you guys are both on the west coast, the rainy part of the world.
That has
Not
been
this time of year.
over 100 degrees the last few days. We've had
For you guys too. Interesting.
Yeah, right.
It's been pretty hot.
Yeah, we've had a heat index of 110 here in St. Louis off and on for the past couple weeks, but I mean that's pretty standard muggy St. Louis weather. I will say that it's struck a little earlier than it has in the past. But yeah, we were usually used to that in July and August, but not June. So that's interesting.
I have friends with no air conditioning, Remy, sitting through 104 degree weather. It's pretty rough.
Nothing's changing though, There's no impact. I wanted to go around the horn and just we're going to talk today about the community science experiment. And I think this is so important. This is one thing, listening to Jake and Mark in previous Reef Therapy episodes, there was talk of these third-party things that should be happening in the hobby, these tests that should be happening in the hobby. We're dosing, carbon sources to our tank, but what bacteria proliferates off of those carbon sources? We're just haphazardly putting things in our tank, hoping that it helps. And what exactly is coming out of that? So we're going to get into that with a little bit of the community science experiment, but I wanted to talk to everybody, see how everybody's tanks are doing. Andrew, you got a really nice one in the background of your shot right now. I don't know if I've cropped it out here in the podcast, but tell us about your tank. Tell us about your system that you've got going on there.
Yeah, it's a reefer, a Red Sea Reefer 750, and it's got SBS kind of up top, and then the sand bed is covered in Ghanies. And it's doing great. It's really, really thriving right now. And it's nice that we're in the summer because I'm pretty busy with my job throughout the year. And then I have a little more time when the summer starts to catch up on stuff that I've been kind of letting go by the wayside. And the big project I'm doing over the next couple of days is I have this huge blue stag that has just grown like 5-gallon bucket size. And the really cool thing about it is that about maybe 4 or 5 inches to the tip of one of the branches is grafted. There's this big green streak, which I think due to the proximity of my Bali green slimer something must have migrated from my Bali green slimer to the staghorn. And so my challenge is to reduce the size of the coral because it's growing out of the water. And it's getting heavy to the point where it's going to collapse onto other corals. And so actually, a la Jake Adams, I went back and watched his video on grafting stags to themselves, which is pretty cool. And he showed how he took some stags that were overgrown in the studio, and he cut off the growth tips, and then he removed the middle section that was kind of less active and taking up a lot of space, and then he put them together, grafted them together, and so it took up less space, but he's able to keep the active growth part of the coral. And so what I'm going to do is I'm going to move that grafted part down to the base, and so that's kind of what you see, and then that big middle part that is literally like the diameter of a 5-gallon bucket, I'm going to take out of my tank and bring it to a local coral farmer so he can frag it up. So I've been thinking about this for the last 3 months, but I haven't had time to really devote a whole day to it. So that's my big project for tomorrow. So
that's really cool. And I think
wish me luck.
Well, good luck. And I think the common phrase I hear in the hobby is, what is it, green fluorescent protein or something, infection. But is that actually happening? What actually migrated over to that coral?
Yeah, so Eli will have something to say about this too, because we've talked about this.
I swear you're baiting me with this whole conversation.
So yeah, so I think that Yeah, so I think that it's some transcription factor that has made it over from the Bali green slimer and has turned on the GFP, the green fluorescent protein genes in the blue stag. So most corals probably have the genes to turn on all those different color fluorescent proteins, but they have to be turned on by a transcription factor. And so I think that's what's happened. I'm not sure how or why, but I think that's what's happened. So Eli, you probably want to say some more about that.
We don't have time. No, I find that hypothesis that has been put forward in the hobby really interesting. But if it's true, there's a Nobel Prize waiting to be won, because that doesn't happen anywhere else in biology. So I'm not the guy to say that cannot be it, but holy crap, if that's it, I mean, that's an unheard of mechanism. I think far more likely, my own opinion, what's probably going on here is it's somatic mutation. Like you said, there's multiple fluorescent protein genes in the genome, and as the cells divide, you could have one part of the coral undergo a somatic mutation, so that one of them gets changed color or gets turned on, and then tissue in that region, continues to proliferate and have that color. I say that because somatic mutation is like a known thing that happens in biology, but a protein level gene transfer event would be just shocking. Yeah.
Where did that start? It's one of those things that just like happened in this hobby where it's like, don't put sarcophytans and acros together, they'll fight. And okay, do we have data for that?
Yeah.
But where did the protein thing even start? Like a protein infection? It's just kind of an odd thing that people say.
I was writing an article about this at one point, like the theories of coral grafting, and I found stuff from reefs.com in like 2005 about it. So some dude just like typed that up one day, I think. I don't think anyone that ever has background experience has ever said anything about this. Because the idea of a protein being infectious, I don't know. I mean, man.
Yeah, it's going through like reverse translation and reverse transcription, and somehow getting into the genome. I don't understand the mechanism.
So you don't think that some transcription factor could at least temporarily turn on gene expression in another coral? I'm not talking about the protein itself infecting. I'm talking about a transcription factor.
The transcription factor is another protein, though, right? That has to still make it out of the cells in one coral and into the cells of another coral. How is an intact protein gonna? I'm not trying to say it's impossible. I'm just looking for an example of like, when does that happen in biology, right? That an intact protein makes it out of one organism's cell and over into its neighbor's cell. If we could come up with some kind of viral mechanism, I could maybe get on board, and say it's a nucleic acid transfer. So we know gene transfer happens, right? Usually with viral vectors or things like that, jumping genes, retrotransposons. That would make me less skeptical. It's when we're talking about it at a protein level that I start going, wow, if that's real, that's like new biology.
I was about to say, what if it's a virus? You need to start doing some viral sequencing. Check each family.
Right.
take any stag and see what's happening.
Yeah.
I'm tempted. I think the idea comes from. Sorry, I was going to say, I think the idea comes from seeing it when we have lots of different corals mixed together in high density. And that could just be, it could not be related at all, but it seems like there's shared colors among corals, but that might not be related.
Yeah. I mean, I can certainly understand how like at an observational level, it's happening, right? You're seeing a blue coral next to a green coral, and the blue coral is starting to turn green, right? I mean, Andy, am I describing your observation correct? You're seeing some.
That's exactly
so
it.
Yeah. So I mean, it's not hard to imagine how that's a striking observation. It makes sense that people would imagine it's the green glowy thing is what's moving. Maybe I'm wrong. Maybe there's some unknown biology here. For now, I am.
It always seems to be green. The color is green though. Usually if
Yeah.
there's going to be any of this like weirdness, it's that lime green kind of fluorescent color that comes through on whatever species of coral it is.
Which is
They talk about this at Top Shelf Aquatics all the time, where they've got the huge farm system, and there's tons of different SPS and different species sharing the same water, and they've had this naturally happen several times, and usually the color is green that comes into the other species. So
that right there is an interesting observation, because if you think about green fluorescent proteins, this is a family of genes that went through evolution, started as a single gene, duplicated, became multiple genes with different colors, and it started green. So what we're kind of seeing is like reversion to the ancestral green color. Maybe that makes it less surprising. Maybe we're getting distracted by the fact that it's green. Maybe it has nothing to do with being next to a green color. It's just a mutation that takes it back to its ancestral green color.
We're distracted
by
your
PhD dissertation, Salem.
What?
There's your PhD dissertation right there.
We're to figure out grafting, people.
Hey, Salem, if you want to do that, I know a very interesting guy to work for who does exactly that kind of thing. Mutate green fluorescent proteins from corals to change their colors. Anyway, another discussion.
It could be like designer. Think about that. Combine that with like what they're doing in Interfish. Create designer corals, like the chimeras, but also like even more colors. That'd be cool.
Yeah,
I don't know. Awesome. Like maybe it's just random chance. Like I think, that makes sense that like GFP would be what it reverts to, or like what the mutation would express. Maybe there's a common mutagen that's in our systems, and that just causes there's a similar pathway between these things. But also, I mean, that's mutation isn't really like conserved like that. It's a pretty random event, but I don't know. That's weird. Yeah, because like there's not any other colors that you really see with it at all. With crafting, there's always green.
That's interesting.
Yeah. Well, that's cool. Let us know how that all goes for you. Would love to follow along on that journey as much as we possibly can.
Yeah, I'm going to try to document it on my Instagram page. I'll take pictures of before and after. And what I'm afraid of is that it's going to all kind of collapse on me. So we'll see.
It'll collapse in front of everybody, and it'll be great.
Well, I'm happy to take some chunks from it when it all breaks into pieces.
Oh, that's good to know, because it's 5-gallon bucket size. It's that big.
Dr. Eli, do you have a tank at home? Do you have a personal tank?
I got more tanks than I should. Yeah, I have one display tank in my living room, just a 90-gallon. But I built a greenhouse in my front yard to grow all the tanks that wouldn't fit in my house. I run a small coral sales business on the side. So I've got a bunch of tanks out there. We'll just talk about my display tank, though. It's the only pretty one. Right now, it's going through a recovery phase. I guess 6 months or so ago, had a pretty big crash, just neglect, and lost a lot of corals. So yeah, restocking and slowly growing everything again.
Can we talk about the coral farm that you have in your front yard?
Yeah. Yeah. For a while I was selling frags out of spare tanks, like I think a lot of hobbyists do. I'd sell them on, Reef to Reef and stuff like that. And early COVID, I guess, it was early 2020. My sales just went through the roof. I was like, man, like everybody wants to buy corals all of a sudden. So I said, okay, let's expand. So I built a 12 by 24 foot greenhouse out in my front yard. Put a bunch of tanks in there and transferred my coral farm out there. It's nothing pretty. All my stuff is kind of budget and homemade DIY, but it grows a lot of corals. Yeah.
How's the HOA feel about that?
So I live out in the boonies. I have cows. I have sheep. There's no HOA anywhere around. Yep.
That's awesome. I just envision like somebody in a subdivision with a coral farm in their front yard. Everybody's out mowing stripes in their yard every week, right. And there's this giant I picture a Than-sized Tidal Gardens building in someone's front yard.
Right, right. No, I just get the farmers driving by wondering why there's these weird blue lights coming out of my greenhouse at night.
That's awesome.
Yeah, they all think it's a weed production facility.
Right, they all think it's a pot farm or something, but no, nothing like that.
That's awesome. That's awesome. Salem, how are your tanks doing?
I'm putting my money where my mouth is, doing my whole DOC thing. So I'm doing 100 gallons of water changes a week. I have ripped out my refugium, never to have again. I added a second skimmer because I got sent one for free. So I'm like, I guess I'll hook them both up. One's just a glorified air pump for my CO2 scrubber, and the other one's just like full force going. I don't really know if that's efficient to have two skimmers or not. Probably not. And then I'm just scraping algae and getting the Tritus out. So I really had not done water changes in like 2 years. I was just running it like full trace element, doing the shine type of stuff, and embraced that. And now I'm like, my ORP is real low. My microbiome is not as good as it could be, that's for sure. I probably need to get some DOC down. And if my ORP goes up, I will send in another test, and I'll see if my hypothesis is correct with a sample size of 1. So I've been doing a lot of manual labor and making a lot of water. And my water bill in Topeka are probably very high. So that's what I've been doing.
Good. Very good. Tanks here. Red Sea's doing okay. Salem and I have been going back and forth on this king spawn that I kind of brought back to life. Got it from Gabe over at Oceanic Gardens. It wasn't doing so hot in his tank. He's like, just take this, see if you can, do something with it, maybe help it out. And in like 2 weeks, 3 weeks, it came back and it was like super fluffy. And then all of a sudden there's like the flesh is hanging off of the side of it. And, immediately I'm like, Salem, what's going on here? What do you think is going on? No brown jelly or anything. No brown. It's like stark white skeleton on half of it. And, I don't know what is your hypothesis? Because I dipped it in just Coral RX because I have it, but you were kind of thinking more of a prosy route, right?
Yeah, that's some secret kind of stuff, kind of. But Chris Meckley and I had some really good conversations, and Chris had some really interesting cross sections of Euphilia, Fimbrophilia that had similar symptoms. There's no no slime, no biofilm, no like external symptoms you would associate with a bacterial thing. And he put them under a scanning electron microscope, and they found microscopic polychaete worms that were boring throughout like everything. And it's almost like an egg, like a larger eukaryotic parasite. So think about how like red bugs, black bugs would affect an individual coral, or like Zoa or Monty eating nudies. You only see one polyp in the colony kind of go down, but it's not a systemic issue. Well, same thing. So Chris and I think there's something eating them. But, we're going to experiment around some. Anything I've seen, symptomatically like that under the scope, nothing. I mean, any of the bacterial stuff, any of the protozoan stuff, fungal stuff, you can see with the scope, and it's just clean tissue. So I think it's a microscopic eukaryotic parasite, either a pod or a worm of some sort.
I have a simpler hypothesis, Remy.
Mm-hmm.
So your Ghani bleached a little bit too, right? In the same period of time. Couldn't it just be a nutrient issue? Because Euphilia will do that sometimes when nutrients go down.
It's possible. Last test was 7.7 for nitrates and 0.01 for phosphates. So yeah, I'm
0.01 is pretty low.
Yeah, and I've been dosing cap full of phosphates, every night or so. And the highest I've gotten it is 0.04.
Yeah, that could be.
But yeah, the colors in the coral are definitely I wouldn't say bleached, but they definitely aren't bold. They're kind of that pale color.
Yeah.
But some of them, everything looks healthy. Like it looks good. It's just like the color isn't necessarily where I would like it to be.
Some corals seem like they're more sensitive to low phosphate. And this is something I've struggled with new systems. My last 2 frag systems, when I set them up, I had very similar issues with corals being pale. and having undetectable phosphate and needing to dose all kinds of phosphate. And when I did dose phosphate, it helped. The corals recovered mostly. So that might be it.
I will say, while that is a likely hypothesis, the way that the tissue looks is not consistent with polyp bailout, at least. It's very like shredded tissue, like, you know, coming off in strips type of thing, which like is so it's not just like the whole polyp, you know, like sometimes they do in a stress event. So, I mean, maybe it's just low phosphates that triggered that for sure, but it's really weird. He showed me pictures. I'm like,
Yeah, well, if you have some ivermectin on the shelf, Remy, you can do
That's what I told him.
Ivermectin dip. I have spinoid or spinoid worms that are burrowing into, they're annelid worms, like little bristle worms that burrow into my SPS, my acros, and that's been pretty effective at getting rid of them.
Is that a whole tank thing or is that a dip?
Oh, it's a dip. Yeah. Yeah, you don't want to put that in. It'll kill all your invertebrates if you put it in your tank. So all your crustaceans are nuked if you put it in your tank.
When I was taking that during COVID, yeah, so that's really the only low nutrients. I've been fighting that ever since, you know, I really started. But you know, there's the SPS all looks great. You know, I've got some choice pieces here from the Reef Builders studio. They're doing great. The Hoaxamai is looking awesome. I've got some of that lazy, the Blue Zing bird's nest hybrid. Yeah, all the fleshy stuff looks really good. So I can't complain right now. It just kind of took me by surprise as I see like half of the flesh of this king spawn just kind of like waving off of the skeleton. So it was doing so good, but that's my only real major issue. That and the Chicago sunburst anemone that I have in my 10-gallon over here decided it was going to split about 3 months ago, which, you know, I was very happy for. I'm like, oh, great, we've got a split. But one half of the split now does the thing that the original anemone did all the time, which was hop up into the overflow. And so now I've had to rescue it from my overflow probably 2 or 3 times. And it's just like hanging in there. And, you know, you got little tentacles hanging out in the tank everywhere because it's like shredded itself as it goes through. And so I've got, just a bothersome nem, a wandering nem. So hopefully that stays put. I'm going on a trip for 10 days. So I'm hoping that everything just kind of hangs out in status quo until I return. So, well, yeah, that's kind of what's going on in my world. So you guys want to talk about the community science experiment? I think this might be the first community science experiment that Reef Builders has ever done, if I'm not mistaken, to this kind of level. But Salem, you want to take it away and kind of tell everybody what we're trying to accomplish with this experiment and where Dr. Eli and Dr. Andrew come in play?
Yeah, so we talked about this a really long time ago, the Microbial Men episode of Reef Therapy back at Reef Stock Denver. We hinted at this, and I have talked about it a little bit, but we've finally got it all formulated, got everything going on. So we now have an official partnership with Aquabiomics. So we'll be using Eli's service to answer community questions. So consider this an audit on the common practices that we have been doing for a long time in this industry. We wish to evaluate what the, effects, or the unseen effects of any of these methods are in our systems. So, going aligned with the dissolved organic carbon stuff, we have chosen to analyze the effect of adding carbon sources on a tank. So we will have 3 test groups. We will have vodka, vinegar, and amino acids. The secondary question we'll answer with this study is how effective the different carbon sources are at lowering nutrients. And then, do aminos lead to an increase in nutrients? So we will have users track their microbiome over a month and also track their nutrients. We've not really decided the frequency of that, but they'll track their nutrients as well, inorganic ones. So hopefully by the end of this, we will have some data to suggest if these practices are best left in the past or there's something we should explore further. So that's kind of what we hope to do going forward with Aquabiomics, is using their service. We will begin answering questions that the hobby has. So likely we'll do a second experiment this year, which we will determine later on, but it will likely deal with nutrient export instead of adding carbon, taking carbon away. So that's what we're doing. We're going to answer the questions everyone has had for a very long time.
So what if I've always just dosed vodka in my system, and that's just what I've always done, and it's worked? What are these, or like you said, or maybe like no pox, or something like that. What is the end goal here? What are we trying to find out? Like, which bacteria is proliferating most off of each carbon source?
Yeah, the goal would be to identify if there's any, conserved families or either in actual structure or function. So, like, if there are specific families that grow or different families that have certain ecological roles, like the same ecological roles that will increase in concentration, or if there's any families that decrease in concentration, and if that is consistent across tanks that are being tested. So, at least I think that likely you're going to have an increase in the copiotrophic bacteria, the bacteria that like carbon. Many of them can be pathogenic. So families like Rhodobacteriaceae, Flavobacteria, Altermonas, things like that. And you would likely see a decrease in oligotrophic or low carbon environment microbes like Pelagibacteriaceae, which we have pretty much determined is likely a very good microbe to have a large population of in our systems. So, we're basically tracking the microbiome because dissolved organic carbon is likely affecting that more than anything else in our systems. I mean, the idea of carbon dosing is to increase the bacterial population. So what bacteria are growing is the question.
Exactly. So, I mean, I've read these discussions on forums for a decade now. We all know that when you add carbon to your system, you're growing bacteria. That's the whole basis for carbon dosing, right? But the debate always came up, which bacteria are you growing? And there was kind of in science, we call it a null hypothesis. I'll call it the kind of a silly idea. The idea has been floated out there that it doesn't matter. That basically when you put carbon into your system, it's just going to grow all the bacteria. I think we're going to pretty conclusively show that's not the case. So I'd say that's the first kind of hypothesis we're testing, is that I suggest it will selectively enrich some groups of bacteria. Some groups of bacteria will bloom, will do much better after we add carbon than before. So that's kind of one question here, is like, does it enrich for some kinds of bacteria? Probably we all agree it's going to, but there is the idea out there that no, it doesn't, that it just makes everything grow. And the other question related is, does it matter what kind of carbon you dose? So Remy, you said you've used vodka. What if a different carbon source led to less of an increase in Vibrio, for example, right? What if we came in and saw that dosing vodka led to an increase in Vibrio, but dosing vinegar didn't? That would be interesting information for the hobby, I think. And I don't know, I can't predict what those effects are going to be, but I think that's the second hypothesis, is that different carbon sources promote different groups.
Yeah, just to jump in, I've heard often carbon sourcing in reef tanks also justified in the idea that it promotes blooms of bacteria that corals can feed on, right? So that's another reason to ask what kind of bacteria we're promoting with carbon sources. Are they bacteria that corals are known to graze on? Are they bacteria that we would want corals to graze on? Those are questions that haven't been answered. So this will provide some insight in that area as well.
I think we're pretty deep into like the science side of the hobby when we're talking about all this stuff. From like a typical hobbyist standpoint, you're usually going to encounter carbon dosing when you've got a tank filled with algae and you're trying to increase the bacterial population to lower nutrients so that hopefully that algae does not grow anymore. That's what the common hobbyist is told. Now with the introduction of aquabiomics into the hobby, we kind of have a new picture. We have a new thing to look at, right? The microbiome or the tank's DNA and things like that. The biggest question that people have with the aquabiomics test is, okay, now I've got this picture of what my microbiome looks like. How can I manipulate this now? And part of this right, this carbon experiment would be to maybe find little, the Pelagiobacter, which we all want in our system, the pink band on the aquabiomics test. How do we proliferate that? How do we make that grow specifically? Or how do we do less Vibrio? Things like that. Am I kind of, is my summation kind of correct there?
Absolutely right. I think at the end of this, we're going to have three new tools that we can use. That is the three different treatments that we are testing in this experiment. Each one of them will now be a new strategy that hobbyists could use. If I need more of the bacterium that vinegar promotes, I now know that I can dose vinegar to get it. Sitting here right now in this conversation, we don't know what those bacteria are, but by the end of this experiment, we will. We'll be able to say, this is what you promote with vinegar, this is what you promote with vodka, and this with amino acids.
So community science is becoming a really important thing for science in general. With the internet, with things like iNaturalist, where people can make observations, everybody has a phone with a camera. You can make observations wherever you are, and you can upload your observations to the cloud where they can be used as data in conservation and recovery efforts. As an example, here along the Oregon coast, since 2013, we had this sea star wasting disease that wiped out like 30 species of starfish, right? And some very important species of predaceous starfish, the sunflower sea star, this 3 foot wide, this huge sea star that feeds on purple sea urchins, almost completely wiped out by this disease over a few years. And a very important part of the recovery has been having citizens make observations all along the California to the Oregon to the Washington coast on the presence or absence of these starfish. And it's important because without these starfish present, the sea urchins overgraze the kelp forest and remove large areas of kelp forest, which impacts the fish community and all kinds of things. And so their iNaturalist and observations of sea stars are a big part of the conservation effort. And so we're doing community science in an even kind of slightly different way, in that we're actually having hobbyists participate in a controlled experiment where one variable is manipulated and the other variables are held constant. And so it's an even more in-depth science experience than most community science that people can participate in. So from a perspective of community science, it's very cool.
Is there
Yeah,
any sorry, go for it.
We're really demanding more of our participants than the typical, naturalist who might be just out bird watching and click a quick picture and send it in. But I think that the hobbyist, the reef tank hobby is ripe for that. There are a lot of scientifically minded people in this hobby who are already conducting experiments, just not in a coordinated fashion like this. To my mind, that's the really unique, special thing about this, is having this kind of coordination where it's not just Salem. You were talking about your N of one with your carbon experience in your tank. There's a lot of those in the hobby, and that's what I think is really special about this, is bringing them together so that we have some statistical power and can actually, draw some good conclusions.
Can you kind of go through the experiment design here? I know that you kind of touched on it loosely, but before the call, we were talking about water changes and how that might affect the experiment, or auto water changes, and are you already dosing aminos? I've heard Salem say that aminos they're not the best thing for my tank, and can you kind of discuss those variables that may impact the experiment?
Yeah. Well, Andy, you've done a lot of data with water changes on your system, haven't you?
Yeah. So I wouldn't say I have a lot of data, but I did collect data on the impact of a large water change on the microbiome. So this was a 30% water change. And I saw that basically over a period of about 4 days, it had a pretty big impact on the microbiome. And so some of the insights that we can gain from that are that if people are doing large water changes, you wouldn't want to do that right around the time that you sample your microbiome. And so just to explain more of what I saw was basically I saw a bloom of Pelagibacteriaceae in the community. So the Pelagibacteriaceae are the oligotrophic or the low nutrient loving bacteria that we like to see in our communities. It's the one that's the most common on the Great Barrier Reef, and it's a marker for healthy systems. So when I did a big water change, about 3 days after the water change, that family bloomed and really dominated the community. And so, an important thing that I should follow up with at some point is what is the impact of a 5% water change, or a 10%? Are smaller water changes as impactful on the microbiome? And so, yeah, so that's what you wanted to know, right, Salem, about that?
Yeah, pretty much. So, I mean,
Yeah.
taking Andy's experience of showing that a relatively large water change can have a immediate impact on the microbiome. For all of our study participants, we think it's likely best to avoid any of those large changes. We would like as much control and consistency with the nutrient input, with the nutrient export, with any of the things they're doing. This is not a time to try something new on your system. Rather, we want you to coast for a month, continue doing what you're doing, stay as controlled as possible. Even if you can weigh out food, things like that, having that amount of control would certainly help the data, I think, go a lot, like be higher quality for sure. So yeah, I mean, potentially if you just did a large water change right before the study, that's probably not something good either. If you're already adding amino acids, we likely, will, I think, potentially just not even have those people take place in the study. I think that made sense what you were talking about, Eli. If they're adding aminos already, then we can consider that they're already adding a carbon source in this instance. It's one of the ones we're testing. So we're looking for candidates that do not run a UV sterilizer because we know that impacts many of the planktonic bacterial families, such as Pelagibacter. And also, we're looking for people who are not carbon dosing or using biopellets already. That gives us clean slates to where then we can have 3 test groups, the 3 different carbon sources, and have 3 people within those test groups, and they can, over a month period, test the effect of those carbon sources on their systems. We will have 3 different tests. So we'll have an initial baseline microbiome test. We'll then go 2 weeks without any changes, do a secondary test as an internal control to show if there's any drift in the populations naturally in their system. And then we will have them carbon dose for 2 weeks, and they will do a final test at the end of the month to show the effect that carbon dosing has on that system's microbiome. Assuming there are no large changes like changing your GAC out or adding Purigen in the middle of dosing the carbon source or doing a big water change. So we want this to be
define GAC for people, Salem.
Oh yeah, I keep saying that. Yeah, GAC is granular activated carbon. So adding some carbon in a reactor, or GFO or Fosgard, any of that stuff. Probably keep things consistent. Don't have a big chemical media change, big water change. Even changing filter floss could even impact things. Maybe take some organics out. Who knows? So keeping things consistent, and we're really looking for this to be, as scientific as we can make it. Lots of controls.
In each one of these tanks, during the treatment period, we're going to be able to directly measure what is the impact on the community, right? So we'll see, this family went up, that family went down. So we'll get that information for every tank. And then we can compare that change to how the same tank behaved in the previous 2 weeks when there were no changes being made, right? So that's the kind of internal control that Salem was talking about. So ideally, we would like to see during the treatment period, you had a family go up, but during the control period, that family did not go up. It stayed flatlined. And so then you can clearly say that the increase in the family was because of the treatment and not just some random sporadic event. Yeah.
Because of that internal control, it's also helping answer a third question, and that is something I've seen a lot is how much does my microbiome change over time? So it also helps answer that question. Maybe there will be a large drift in some tanks. We don't know. I suspect likely not if there's consistency in the husbandry, but we'll see. So there's multiple things we're answering with this study for sure.
And just to follow up on that, Salem, I did a control non-manipulated microbiome to compare my water change my water change data with. And when I just measured every day with no intervention, it stayed pretty much the same. So we have a small sample size of that. But yeah, this will be great. We have a lot more.
Yeah, I get this question a lot, and that is often my answer. That is usually my experience, that typically the community stays pretty stable, and if you measure it repeatedly, you will typically get very similar community measurements. But boy, we've got a few tanks in my data set where it's wild how much it changes from one sample to another. And when I talk to those guys, it's like, well, yeah, they were doing all kinds of stuff during that period. So it's my answer about how stable is your community really has to be tempered by that. It's like, well, it can be very stable, but it is also very responsive. You know, the smallest things you do to your tank, taking your refugium offline, boy, that's going to have a massive impact on your on your. You
I
know,
hope
and
so.
that's just like removing something from the system. You know, that's not dumping new nutrients in. So yeah, it can be stable, but it can be very responsive too. I hope that our participants, you know, take our instructions to heart and keep things really stable during the month.
And just to add on to that, one of the things that I think is really interesting that Eli has discovered with testing all of these tanks is that as tanks mature and certain families show up, specifically the Pelagibacteriaceae, they become more stable. And so if somebody had a brand new system, say 3 months old, we would expect it to bounce around a lot, right, from sample to sample. But my systems, 9 to 10 months in, once I have a dominant Pelagibacteriaceae family or present in the microbiome, they tend to be pretty stable from sample to sample.
So I have a maybe kind of an ignorant question here from the outside of science. Are we dealing with bacterial families that only exist in a marine environment? Are some of these families also present within us? And how do we not know what bacteria feed on, like what makes certain bacterial families proliferate more than others from a feed, from a carbon source standpoint? I don't know if you can answer that, but that's kind of where I'm like, how do we not know this? Don't we have some
Yeah,
sort of research like in our
yeah,
own gut?
right, right. I'll take a stab at that. So, um, the amount of salt present in an environment is one of the biggest determinants of which kinds of bacteria can live there. Um, so you'll find very similar bacteria in, say, a freshwater environment and a marine environment. But if you get specific enough and go down to the species level or even the variety level within a species, you'll find they are different. It's quite rare to have bacteria that can live across a wide range of salt levels. So that's the biggest thing that makes me say no. The stuff we're finding in our tanks is by and large not the same stuff that we find on our tables or our skin. You know, there's a few exceptions to that. You find a few bacteria that cross those environments, but for the most part, no. How do we not know this stuff? I mean, I love that question. The fact is, like, we don't know this stuff for most bacteria on the planet. There's whole families that we didn't even know the family existed until a decade or so ago. Whole bacterial families, the Pelagibacteriaceae, this one we're talking about, probably the most abundant bacterium on Earth because it thrives in the open ocean, which is like the biggest part of the biosphere. Until we started sequencing DNA, we didn't know it existed because you can't, or you could not culture it. And so if you don't know it exists, you really can't make any progress in doing experiments on, you know, what does it eat and that sort of thing. That now I've just given you an extreme example, but that's like an extreme famous example. And of course, there's millions of others that we don't even have names for them yet, let alone have we done the experiments to know what they eat. Yeah, there is a huge amount unknown in this area. We can make some generalizations, right? If you go up to the genus or family level, you can start to make some generalizations and say, well, this genus tends to do these things. And so even though I don't know the species, I can assume it's going to behave like other members of the genus. the farther you get up on the taxonomy, the less reliable those generalizations are. So I think my best answer is, yeah, there's just a lot unknown. That's just the state of it.
That's fun, right?
Yeah,
That's kind
yeah,
of
it is.
fun.
Absolutely. Yeah.
But I think you know, when people are when hobbyists are throwing around these names like Vibrio, that's a family of, right. So there's a lot of species within that family. So you can't just say, oh, it's Vibrio. It is, but what specific kind of Vibrio?
Yeah, yeah, yeah. We know that there's so I mean Vibrio. There's the family of Vibrionaceae and the genus Vibrio within it, and there's lots of kinds of Vibrio within that genus. Some of them we know are normal parts of the coral microbiome. They exist in the coral mucus. They're a healthy part of that community, and of course, others are terrible pathogens for corals or even for humans. Um, yeah, so it does differ as you get down to the species level. But one thing you can say at the level of Vibrio is, boy, this is a genus that has a lot of pathogens in it. If I see this genus at a really high level, that's probably not an ideal thing.
Yeah, that's something that's really important when looking at all of this data. If you do an aquabiomics test or you're beginning to read the literature, is the function of these families generally. So, like a lot of systems I'll see that have, higher nutrients, things like that, or they've never really added any live rock. They're typically dominated by families that are kind of filthy families if you look at how they live and the types of environments they thrive in. So the same ones I mentioned earlier, you'll see things like Flavobacter or Rhodobacteraceae. Both of those families to the untrained person they're completely different. But in the environments they grow in and the types of conditions they both like really high nutrient kind of dirty environments. So something also to think about is this is an ecosystem we're looking at. So think about the roles that you would have in an ecosystem. There's scavengers. There's kind of, there's pathogens. There's ones that cause disease that are kind of like a predator. So thinking about things in those kind of analogous terms, it's really it's a microbial ecology question, and looking at the function of these families is extremely important, I think.
Yeah. I wanted to ask earlier. I remembered my question, and that was, are you guys? I know that there will be only a few spots to get the tests for free. Would you guys allow it if someone could self-fund getting those aquabiomics tests and also participate? Because I want to say, what would that end up being in the neighborhood of like $300 or so to get those tests?
Yeah, I mean less because we could do the quantity discount on them. Yeah, this is a good point. I'd be open to that. I mean, um, as long as they're willing to sort of play by the rules and follow the same, protocol that everyone else is. A higher end that people want to fund themselves, I don't see a big downside. What do you guys think?
Yeah, I think we just need to have three rules: is follow our rules. Second, you'll be provided a carbon source, so don't just do your own, I suppose. And I think that makes sense to build the sample size of what we're testing. And then third, we need to probably have a deadline for when these people can. I guess not. I guess they could do it whenever if we have the carbon sources available. It can always be a continual thing we add to. So maybe not a deadline. But yeah.
And just to add, if there's a lot of people out there that are interested in participating in the future, perhaps they don't make the cut for this one. We'd like for this to be a recurring thing where we test other things. So we could learn a lot about the microbial community in our tanks and how to change it, and using as Sanjay likes to refer to it, a million monkeys, right? All the people that are out there that have their own tanks that are able to do experiments for us. It could be a very powerful thing for the hobby.
Well, while we're
Yeah.
speaking to potential participants, let me just say again that in my view, the most important and maybe the most challenging thing is having the restraint to not do anything different to your tank for a whole month. I mean, if a fish dies, you don't medicate your tank. I mean, if you get an algae outbreak, you don't stop the experiment, right? That's hard. That's something that we should make sure we have that discussion with every participant. because if you stop halfway through, then your first samples are of no use.
Yeah. I wasn't going to mention the whole like self-funded thing, but then I thought about it and I was like, how many hobbyists aren't spending like $300 a month on their tank anyway? You know what I mean? And you could say maybe I'm on a budget or whatever, but I saw you buy that torch and that other torch and that Acropora. Like, I know you're spending money, but if you want to help the community, I think this is a great way to do it, right?
Yeah, that's a very good point. Yeah, this was the limit of what I could fund, but if other people want to fund it, jump in.
Yeah, this is an opportunity to be a part of history. This is reef history happening right now. No one has really gone and just said, let's look at like what's happening under the hood if we do this to our tanks. I think this could be, the beginning of something very big to where we can really determine what methods are the healthiest for our systems and can usher us into a new generation of reefing where we design products or, design methods around what we have data to support instead of, X company said to add this, it's got to work, and not questioning anyone. I think that transparency and what's in the products and what they can do is important. And if we're being honest here, the companies aren't going to do that work. So community science project, little community whistleblowing, transparency being forced.
Yeah, I think BRS Investigates back in the day, attempted to do a lot of this, kind of experimentation on different tanks and things like that. But even still, I mean, they might have had 10, 15 tanks that they were testing on, but it was all in the same facility. It was all the same, kind of how we make the water. Now we're looking at, how hobbyists do things on this spectrum. It's like and I've seen a lot, and you guys too have probably seen the down and dirty, tanks. How are these tanks surviving kind of thing to pristine waters, know all the parameters, there's that spectrum. And I think when you're going out into the community, you're getting a better picture of what works and what doesn't. And like you said, Salem, I feel like this is just the beginning of something very awesome. Like, I've been thinking about this for years. If we all put our heads together and figured out how to spawn corals in our home tanks, like what then could we do, to make that happen in the ocean more often, or save corals, and for lack of a better term. Obviously there's biosecurity things in there, but what are the things that we can use? People are doing chemistry and biology stuff every day, whether you know it or not. Like, you're doing that. And I think that we can use that data to our advantage for sure.
Yeah, like you said, there was an attempt at getting some answers, but this ought to, I think, give us some real ones. So this is much better designed than previous attempts, I would have to say. So this is different.
Well, replication is key. I mean, every early grad student learns this. You can run a experiment with 1 or 2 replicates and get really excited about what you think you've discovered, and then you run it with 5 replicates and discover that no, that was just spurious results, not reproducible. We're aiming here to do reproducible science, where the conclusions we come to will also apply in your tank. And the boring detail of replicates is what achieves that. Having multiple people do exactly the same thing. Yeah. That's often been lacking in the hobby.
Yeah. And just like in science, small studies, pilot studies, often lead to different questions that lead to other projects. And so this very well could, springboard into a different study based on what we learned from this one. So there's a lot of possibilities here.
I think also, because this is a consumer-driven project, you should comment below what other things you would like to evaluate in the future. And we are open-minded to testing just about anything, I'd say. We had some very interesting ideas of things to test that are not too common, that are kind of cutting edge. But yeah, we want to see what you all want to know, and we will try to answer those questions.
Very good. Do you guys have anything else you want to talk about with this experiment? I would like to obviously, say if you want to reach out to who would be the best person. Salem, would you be the best person, the point person here?
Yeah, I can probably coordinate everything and get some emails, or I'll just create an email chain. That's also something I can do. If there's like, someone wants to really buy some bulk test, something like that, I can redirect you to Eli, obviously. But emails will be sent out immediately after this podcast. So if you get an email, you have been selected, or you're in the final selection stage for the experiment. So check your emails if you filled out my Google form on Facebook like 7 months ago.
And we'll do our best to we'll be available for consultation. So if you have questions about what you think you should or should not do, we're happy to answer those questions. So I don't think just nobody should be intimidated by this. It's going to be pretty easy to do for the average person, I think.
Very good. Well, I want to thank Dr. Eli, Dr. Andrew, Salem. Appreciate you guys for being on today's session of Reef Therapy. If you want to sponsor, so we're still sponsorless on the Reef Therapy podcast. Remember, crime number one is that you got to be cool. If you're not cool, you're not coming on. Want to thank you guys for coming on, and we will catch you in the next one. Bye, guys.
Right.
Thanks.
Thank you.
See ya.
Bye.